Hello everyone, and welcome to the special investor briefing for the collaboration between AdAlta and Carina Biotech that was announced to the ASX yesterday. My name is Jane Lowe and I'm the Managing Director of our department, and I'll be your host for today's session. Presenting today will be AdAlta's CEO and Managing Director, Dr. Tim Oldham, as well as Carina's Managing Director and CEO, Dr. Deborah Rathjen. Accompanying Tim and Deborah will be Professor Shaun McColl, who is Vice President of Carina's Chemokine Receptor Platform, as well as Professor Mick Foley, who is AdAlta's Chief Scientific Officer. The formal part of this session will be followed by Q&A. If you would like to ask a question, you can do so at any time by entering your question into the Q&A function in the Zoom ribbon at the bottom of the screen. And with that, I'd now like to hand over to Dr. Tim Oldham of AdAlta. Thank you, Tim.
Hello, everyone, and thank you for joining us on this very exciting day for Carina and AdAlta. In 2012, a 6-year-old girl in America had run out of options for her leukemia. She'd been treated with multiple rounds of chemotherapy, had failed a bone marrow transplant, and her doctors had essentially run out of options. They tried one last technique, and this young girl became the first person ever in the world to have her immune cells removed from her body, engineered in a laboratory to fight cancer, and returned to her. In May 2021, she celebrated 9 years cancer-free. This therapy is what is now known as CAR T therapy. The ability to engineer a patient's immune cells to fight their cancer offers the potential for a one-time treatment that is potentially curative, is certainly long-lasting, and can be very highly targeted. CAR T therapy is revolutionizing cancer treatment already for thousands of blood cancer patients around the world, achieving up to 90% complete remission from their disease. Today, we are delighted to announce Carina and AdAlta's additional contributions to this exciting field. We aim to combine AdAlta's unique iBody technology with superior tumor targeting capability with Carina's world-class CAR T technology platform to generate next-generation CAR T cells capable of improved targeting, improved tumor access and potency, and increased functionality. Our aim is to offer new hope to the 90% of cancer patients out there today with solid tumours. We will be making forward-looking statements today. This presentation is being recorded and has been lodged with the ASX, and it is subject to the usual disclaimers. And with that, I'll hand over to Deborah Rathjen.
Thanks, Tim, and hello, everybody. It's our pleasure, our great pleasure, to be partnering with AdAlta to really develop precision-engineered CAR T therapies that will provide new hope for patients with cancer. Over the past 5 years, Carina has developed a powerful CAR T platform that is capable of very efficiently generating CAR T cells with superior vitality, potency, and ability to access and kill solid tumours. Our goal is to bring this amazing technology to more patients as we help create a future that defeats cancer. We've been evaluating technologies that offer superior targeting and a way to produce bispecific CAR T cells to target multiple tumour antigens simultaneously, something that we believe that AdAlta iBodies will enable us to do. This graphic explains how CAR T therapies work. The immune system is finely tuned to target and eliminate pathogens. The ability to eliminate pathogens is based on unique structures called antigens that are found on every cell in the body. Unfortunately, cancer tricks the immune system and is able to look like any other normal cell in the body, and it's very adept at hiding from the immune system. CAR T therapy turbocharges the body's own immune system and its response to cancer by engineering a special kind of immune cell, a normal cell that is found in blood called a T cell, so it can see cancer. As a first step, T cells are collected and isolated from a patient's blood. In the laboratory, then they're then very precisely engineered to express a new receptor called a chimeric antigen receptor, or CAR. Now, this CAR specifically recognises antigens that are found on cancer cells. At Carina, we've learnt that you need more than the CAR. Carina's technology also enables us to add other features called chemokine receptors that improve the ability of the CAR T cells to leave the blood and to penetrate a solid cancer and remain active within the cancer. These newly created engineered CAR T cells are then expanded and given back to the patient, where they can now hunt down and eliminate the cancer. So why is CAR T therapy so powerful, and why are we so excited about it? A single dose of CAR T cells, even in patients whose cancer has resisted multiple rounds of standard therapies, and for the first time, oncologists are beginning to talk about cancers that are curable. CAR T therapy can be long-lasting because it's a living therapy. It's a living cell therapy where the CAR T cells are capable of surviving and replicating, so that if cancer recurs, they're available to, again, repel the cancer. And so, for an extended period of time, we believe CAR T cells can provide a signal and an efficacy that really is better than current drugs, better than current treatments, which need recurrent administrations. Now, CAR T cells are precision engineered. They're very highly targeted, and they specifically engage with cancer cells. They do not engage with normal healthy cells. And this is why they're able to bring about fewer side effects, and unlike chemotherapy and radiation therapy. Our collaboration with AdAlta is really very simple in concept, but it's very much at the cutting edge of cancer medicine. We have our world-leading proprietary technology, which gives CAR T cells superior access, superior potency, and greater resilience within the tumour microenvironment. AdAlta brings its unique iBodies, which are obviously able to reach a greater, broader group of antigen targets that have exceptional reach. And in our collaboration, we're going to particularly focus on next generation CAR T products that are either bispecific or dual targeted. Now, what does AdAlta bring to the collaboration? AdAlta is really targeting the antigens. They're bringing specifically designed iBodies that will be able to address either difficult targets or intractable antigens. They're half the size of a traditional CAR, which means that they're ideally suited to this bispecific or dual CAR approach that our collaboration will focus on. And, we've already made a great start. We've got in vitro proof of principle around one of the established AdAlta iBodies using the Carina CAR T platform. Carina brings to the collaboration and will benefit from the collaboration through a number of different features. I'll highlight particularly our industry-leading CAR development efficiency, our ability to not only create high-efficiency CARs, but also to reduce manufacturing time. We're really at the leading edge of best practice with regard to industry manufacturing of CAR T cells. And then the unique features of our chemokine receptor component of our CARs, which enable the homing of CAR T cells to solid tumours and provide the direction they also enhance CAR T cell killing. So the combination of these two features, our manufacturing ability, which is fully integrated within the process of our CAR T generation, and also this homing capability of our chemokine receptor component, really brings about CAR T persistence and gets us a lot closer to that single shot therapy that we're aiming for. If we go and look at what is the difference between a normal CAR, a bispecific CAR, and a dual CAR that targets specific cancers. A dual CAR has two iBody components attached to it in two separate CAR engineering systems. A bispecific CAR will have a single CAR, but with two components, each addressing a separate antigen. So whichever way you look at it, whether it's a dual CAR or bispecific CAR, it will target at least two antigens that are present on cancer. There are a number of advantages to this, and one of them, of course, is that it reduces the opportunity for cancer cells to be missed. And then the second one is, again, you're reducing the potential for any off-target activity, any off-cancer activity, so that you've got a reduced chance of hitting a healthy cell. So we believe this brings a powerful new element to CAR T technology.
So what's the enthusiasm commercially for this space? The CAR T therapy market is a new market, but it really does have enormous potential. When the first two CAR T products for blood cancers were approved in 2018, they garnered headlines along the lines of the greatest advances in cancer therapy in a century have arrived. By 2020, those first two products were already generating more than $1 billion in sales a year. Overall, the market is expected to grow at more than 20% a year from here as new therapies become available to increasing numbers of patients, and in particular, as we start to achieve CAR T therapies against solid tumours, which represent 90% of all cancers. Recent forecasts suggest that the overall CAR T market will reach $20 billion by 2028, and that by 2030, the revenue from solid tumour CAR Ts will overtake that of blood cancers. This slide summarises the 5 FDA-approved CAR T therapies that are so far approved for blood cancers today. Of particular note is the size of the transactions that have been associated with these early products. And note that the big ones were all before the products were even approved. The almost $12 billion that Gilead paid for Kite Therapeutics and the almost $9 billion that Celgene paid to acquire Juno represents the transformational value and highlights the transformational value of these therapies and the value of a robust CAR T pipeline. There is a continuing, very active deal space for CAR T therapies at all stages of development, from proof of concept all the way through to clinical, with big pharma and biopharmaceutical companies very active in acquiring CAR T cells. The strategic fit for this collaboration is really well balanced. For Carina, it clearly builds on Carina's existing expertise in precision-based cell therapies. And as Deborah has outlined, access to the small format iBody really enhances Carina's aspirations around bispecific targeting to really create those unique CAR T cells we're going to need for accessing and addressing the challenges associated with solid tumours. In addition, this enables Carina to have a far more aggressive and greater control over the expansion of its own pipeline. For AdAlta, the strategy we've always had is to use, or one of the strategies we've always had is to use the iBody to help deliver another therapeutic cargo. And in some respects, CAR T therapy is just another example of that. We've always thought that the iBody would have advantages over the traditional fragment antibodies used to target CAR T cells, especially in this context of dual and bispecific CAR Ts. But we needed a collaboration like Carina in order to be able to do this. So it really is very much a marriage of the two complementary capabilities to create a product that neither of us could have created together. This collaboration is the second example of a co-development collaboration of this kind, adding to our existing collaboration with GE Healthcare, and it contributes multiple products to our pipeline. Turning now to the way the collaboration will work. The initial collaboration will be on up to 5 tumour-specific antigen targets. We've selected the first 2 of these, although we will not be disclosing their identity today. It is possible to combine these targets to create bispecific or dual CARs as well. AdAlta's role is to discover a panel of iBodies with a range of binding affinities that can be used against these targets. Carina will then combine these iBodies into their CAR T cell platform to create a panel of CAR T cells from which we can select an optimum product candidate. The companies will then jointly fund the in vivo mouse studies to demonstrate proof of concept. The parties will jointly own the collaboration IP that emerges from this proof of concept stage, and will have multiple commercialization options post proof of concept. We can elect to continue to co-develop the products together. We can choose one or either of us to progress each individual product, or we can out-license each individual product immediately. And as I mentioned, there is a very active biopharma deal space in this area, and large biotech and pharma companies are actively sourcing CAR T products. So what are our next steps? Well, we're actually well underway. In the exploratory work prior to announcing our collaboration, we've established that it is possible to insert an iBody into a CAR T cell and have that CAR T cell be capable of killing a target cell expressing the antigen that the iBody was directed against. We've also selected the initial 2 targets. Yesterday, we announced our collaboration and formalized our arrangements together. We are now finalizing the research project outlines for those first 2 targets and expect to commence iBody discovery in the next month or so. We'll also continue to work on the selection of further targets. By the first half of next, or in the first half of next year, we expect to be able to communicate and report the first experimental results in CAR T cells on these initial targets. So you can see our contact details here for further information. But to close, really, it's a thrilling day for us at AdAlta and our partners at Carina to announce our collaboration. We've both seen opportunities to advance CAR T therapy into solid tumours. We both have parts of the puzzle, and together we believe we have a unique collaboration that can generate a pipeline of precision-engineered, iBody-enabled CAR T therapeutics that are going to create new hope for the 90% of cancer patients that suffer from a solid tumour. And this is very much the beginning of what we expect to be a long and fruitful collaboration. With that, I'll close the formal part of our conversation, and we look forward to addressing any questions that you may have.
Thank you, Tim and Deborah. I'd now like to open up the session to questions and to invite Deborah back on screen. Thank you, as well as Professor Shaun McColl and Professor Mick Foley. Okay, and we'll kick off with the first one, and I might direct that towards Mick. So if you can please just talk us through, why did you decide to pursue CAR T as an application for iBodies, and why did you select Carina?
Yeah, so just echoing what Tim said earlier, one of the strategies is to use iBodies as targeting vehicles to help deliver therapeutic payloads to desired locations. And so using them to direct CAR Ts to tumours is really just another example of this. You know, the iBody packs a very tight binding, high specificity into a very small sized antibody fragment, meaning they can access targets that traditional antibodies can't. And as you heard, CAR T therapy is revolutionizing cancer outcomes in blood cancers, but that hasn't really translated into solid tumours. And we believe that the iBodies offer an advantage over traditional antibody fragments used to target CAR-Ts at the moment, particularly, for example, the ability to access unique tumor antigens, easy to tune or to optimize the binding affinity for optimum CAR-T cell killing. And in particular, as a result of the small size, fit more than one iBody into a CAR-T to create bispecific or dual CAR-Ts, which are emerging as a very important strategy in solid tumors. And as to why Carina, well, we wanted to partner with someone who had world-leading CAR-T technology, and we were impressed with Carina's manufacturing process, particularly the high efficiency. So, for example, they can get 90% of starting T cells converted to CAR-Ts. Expansion takes only about 9 days. And we like Carina's focus on maximizing potency so that the T cells are not already exhausted at the end of manufacturing. And I guess finally, importantly, I think we've already worked with Carina to produce an iBody-enabled CAR-T and demonstrated that it's able to kill target cells expressing the antigen that the iBody binds to. So we're very excited about this collaboration.
Thanks, Mick. Okay, perhaps one for Shaun now, approaching it from the opposite direction. So why did you select AdAlta to pair up your technology with, Shaun?
That's a really interesting question, and I think Mick's demonstration there of how well he understands CAR-T cell therapy on top of the iBody approaches indicates quite clearly one of the reasons we're excited about collaboration with AdAlta. Look, you've heard from Deborah quite clearly that we have developed a powerful CAR-T cell platform that's capable of very efficiently generating robust, highly potent CAR-T cells with superior ability to access tumours. Our manufacturing process is, it's actually leading industry best practice at the moment, as Mick just pointed out, very short manufacture time, highly efficient transduction efficiency, and this will greatly reduce cost of manufacture, which is an important consideration in CAR-T cell therapeutics. We've been working actively on enhancing the ability of CAR-T cells to enter tumours and fight the tumours. So we have technology that allows us to achieve that. And along those lines, we've already produced a potent CAR-T cells with a target antigen, LGR5, soon to be tested in a first clinical trial in colorectal cancer. So we're very excited about the potential moving forward. But solid tumours are incredibly complex, and because of this, they're a big therapeutic challenge. That they are heterogeneous, and most therapies also drive tumour escape. So for this reason, while CAR-T cell therapy offers greatly improved specificity, targeting multiple tumour antigens simultaneously is likely to be required to be effective against a range of solid tumours. In other words, treating multiple cancers will require multiple CARs. So AdAlta's iBody platform offers a number of outstanding features that attracted our attention, particularly related to what I just said. The iBody technology is very powerful, combining high-level specificity, and because iBodies are so small, it also gives us the opportunity to create CAR-T cells that target multiple tumour antigens simultaneously. Related to this, because of AdAlta's world-leading capability in screening to identify and produce iBodies, the partnership provides the opportunity to identify many additional tumour antigens to make new CARs, further enhancing simultaneous targeting of tumours. And you've heard us talk about dual CARs, bispecific CARs. This is exactly what the AdAlta technology is going to allow AdAlta and Carina to bring to patients. So together, Carina and AdAlta will have a powerful toolbox to create next generation CAR-T cells to extend their application in the treatment of solid tumours.
Thanks, Shaun. Perhaps one for Deborah now. So there's an increasing amount of press about CAR-T therapy. What is it that sets your technology apart?
Thanks, Jane. Well, our technology is set apart partly because we look at all of the different elements that are required within the technology to be successful. We look at the antigen targeting, and in our own pipeline programs, we've chosen very selective targets that have a high level of specificity for cancer. And a great example Shaun's already mentioned, which is the LGR5 target. Now, LGR5 is a well-known cancer stem cell target. It is known to not only initiate cancer, but also to be absolutely needed for cancer spread in a number of different cancer types, including colon cancer, ovarian, and it's also associated with the most aggressive forms of neuroblastoma. So we've been very interested in differentiating the antigens that we go after, and the AdAlta technology, the AdAlta iBody technology, is a great fit with this philosophy and strategy for Carina. Then the other element is the work that Shaun and his group have done now for, well, close to 30 years around important chemokine biology and their role in, and chemokine receptor biology and the role in the immune response to cancer, and now applying that to CAR T technology to ensure that we have really resilient, high-powered CAR T cells that are not exhausted by the time they get to the tumour, and that they're able to be available either within the tumour or within immune organs like the spleen should cancer recur, and be able to be activated again to fight the tumours. And we've had some really fantastic preclinical results with the LGR5 CAR. As I said, you know, it's about three components. It's about the ability to recognise the cancer in a way that's differentiated from normal cells. It's about getting the cancer cells there in a state to really be actively fighting the cancer. And then it's about making sure that the process of making the CAR Ts is best practice. One of the big challenges has been to get that time down so that firstly patients can get their CAR T cells sooner, but also you're reducing the cost of goods. Now, some of these therapies, obviously, as you saw from the slide that Tim presented, are very expensive. So by reducing the time of production whilst maintaining and even building on and improving the quality of the CAR T cells that come out of the process, then we're able to really deliver on each of the challenging components around CAR T therapy. So I'd say that those are our differentiation features. It's not one single thing, but it's how we combine all of those as we move through our process.
Great, thank you. We've had quite a few questions come through on the commercial framework of the collaboration. So I might address these to you, Tim, in the first instance, and then perhaps Deborah can jump in as needed. So first question is, looking for some extra details, what are the trigger points for commercialisation decisions? And then I've got a few more for you, Tim.
Thanks, Jane. So the really first milestones that we're trying to achieve with this collaboration is what we're calling in vivo proof of concept. So that will be a range of CAR constructs that have been used to treat mice and demonstrating significant and improved tumour reduction relative to other options. So that's really the first point at where we think we've got proof of concept, and that becomes really our first decision point around licensing a product. And at that point, that's the big trigger for us to make a decision jointly as to how we want to progress that. So that's really, I guess, the sort of first milestone and decision point that we would be seeking. Well, obviously, we have interim milestones along the way from an internal development point of view, but that's the first outward focusing trigger point we would see.
Thank you. I've got a question here just around sort of funding of the collaboration. And the question is trying to get a sense of what sort of cash investment this will require for AdAlta over the next 6 to 12 months. Mentioned joint funding of mouse model studies. Is this a 50-50 split?
So the overall collaboration is structured on the basis that, yes, we think the costs of getting to that in vivo proof of concept stage are equally shared. Now, AdAlta bears the cost of the iBody discovery, Carina bears the costs of CAR creation and in vitro testing, and we jointly share equally the costs of the mouse studies. From an overall perspective, those costs are not going to be that different to the collaboration. To any of our own internal discovery programs, where we'd still have to discover an iBody against a target. We'd still have to screen it for its biological functionality. In this case, we're sharing the costs and sharing the IP that comes out of it. We have stated since we launched our new strategy in March last year that we expected to add and have up to 5 programs in our pipeline by the end of this year. We've continued to reiterate that guidance, and that one of those would be in a collaboration of some type. And this is the collaboration we've been referring to. So in that context, all of our current cash flow projections reflect our expected contributions to this collaboration.
Great, thank you. And finally, just one left here for you, Tim, is on IP ownership. And Deb, again, feel free to jump in as required. So how is IP ownership being handled through this collaboration?
So each party owns their background IP. So Carina will retain full control of the Chemokine receptor platform and their manufacturing know-how. AdAlta retains full ownership of the iBodies that go into the collaboration, and we jointly own the IP associated with the specific CAR products that emerge from the collaboration. And so that means that, yes, when we choose how to proceed at the end of the in vivo proof of concept, we jointly own the product equally, and we share the rewards and returns equally. Now, bear in mind that we have multiple ways of collaborating from that point onwards. If we out-license, then it goes to a third party and we will share the returns equally. We may choose to appoint one or other of us to pursue any individual product. And bear in mind, we can make these decisions on a product-by-product basis. At that point, we've actually pre-agreed a large part of the terms under which we would license our share to the other party and vice versa, if that was the way we went forward. And of course, we are reserving the right to progress the co-development of those products together, in which case we would continue to share costs. So the way it works is essentially that the Carina and AdAlta have a right of first refusal to progress a program past that preclinical proof of concept. We both have options to take on the development ourselves. And if we choose not to, or choose not to co-develop, then we would seek to out-license.
Thank you.
Thanks. And Tim, if I could just jump in and add that all of the different scenarios around which way to go in the collaboration have been fairly carefully thought through and are incorporated into the agreement. So we've already discussed what could happen under different scenarios and agreed a path forward in principle around those arrangements.
Thank you. Okay, Mick, I've got a fun question here for you. This is from Stuart Roberts. Thank you, Stuart. And he's interested in knowing, and then I've got a serious question for you. How do you feel about your beloved Celtics this year? Oh, you're on mute.
Yes, I saw that. Thanks, Stuart. Stuart knows I'm a big Glasgow Celtic fan, and this year we've got an Australian coach, Ange Postecoglou, and he's doing wonders, and we've got Tom Rogic. So I'm terribly excited about this year. Celtic will again be the greatest team probably on the planet. And I'm as excited about Celtic as I am about this collaboration, I would say, Stuart. So.
You've been media trained, Mick Foley, that's great. So can we just come back to a science question for you? How do you plan to drive infiltration of CAR T cells into solid tumours? And if you need to share that with Shaun, please go for it.
So probably Shaun's better placed to do this, because I think that's the key for their Chemokine platform, and he's much better able to explain it. I mean, our iBody will be able to bind with high affinity to the tumor target, but increasing the infiltration into solid tumors is Shaun's expertise.
Yeah, so I'll follow up on that. So as Deborah pointed out, we've been studying the molecular control of lymphocyte trafficking into peripheral tissues, and you can consider a tumour, solid tumour, to be generally in peripheral tissues, because they normally begin from epithelial cells which line peripheral tissues for the last 20-30 years, and the group of molecules that we study are called chemokines. And they're called chemokines because they combine two important aspects of the immune system. One is that they're cytokines, gene products, proteins that are released from cells and act as messengers, with a very important function called chemotaxis, which is the chemically induced migration of cells, so the chemicals being these cytokines. So we've been matching up the chemokines that are produced in tumours to receptors that we know bind these chemokines and respond in terms of cell movement with our CAR T cells. So we've been manufacturing CAR T cells that reflect what the tumours are producing, and this enhances the infiltration.
Thanks, Shaun. That's great. And I think you may have touched on this earlier, but perhaps I can sort of just ask you to run over this one again. So can a bispecific iBody, perhaps this is a Mick question, we can figure it out, engage two tumour antigens simultaneously? Or is steric hindrance an issue? Is simultaneous binding required for CAR T cell activation?
Again, I think that's probably Shaun, because it depends very much on the format that you put these into the CAR Ts.
Yes, so look, I'd answer that as being a very good question to start off with, a very sophisticated question. The point I would like to make here is that we have the capability to test exactly that in vitro before we'd ever do any in vivo testing. So we could manipulate and modify the CAR, the bispecific CAR, to make sure that steric hindrance is not an issue.
Terrific, thank you. Okay, question for Deborah. Hi, Deborah. What is your current predicted timeline for entering clinical trials?
We anticipate that the LGR5 CAR T will enter clinical trial next year. We're working towards that plan very strongly.
Thank you. Okay. This might be a question either for Tim or for Mick, and on AdAlta's technology. So AdAlta's now got AD214, which is in development for lung fibrosis, then a collaboration with GE Healthcare for PET imaging, and now this deal focuses on CAR T therapy. So that's a pretty diverse range of uses. Just how broadly can you use the iBody platform? Tim, do you want to start there?
Thanks, Jane. Yeah, it's a really good question. I mean, AdAlta's iBody technology is explicitly a platform technology that's going to have multiple applications. We also know that we can't possibly exploit all of those applications in-house ourselves. So our core expertise and the core capability that we deploy in all of our product platforms is our ability to find iBodies with exquisite selectivity and ability to target specific antigens in a particular way. For our internal program, we focus on a class of receptors called G-protein coupled receptors. They're a very important drug class, but antibody drugs have really struggled to address these well, and we think iBodies are particularly advantaged in this space as a therapeutic agent. And AD214 fits into this category. And so our internal pipeline is going to continue to focus on this family of receptors and continue to focus in the field of fibrosis, inflammation, oncology, because that's what we're learning through both our internal program at the moment and our external collaborations. For other applications of iBodies, it's really important that we have partners like GE Healthcare and now partners like Carina, because that's what brings the complementary technology to the party. We could not hope to deliver a CAR T therapy on our own without building the know-how from scratch. You know, Carina are delivering 10 years of fundamental research into how to solve particularly these solid tumour problems, and 5 years of success and at least 3 products in their pipeline already, several of which have already been out-licensed. You know, what more could we ask for to accelerate and broaden the range of our iBody targets? So we can be very, very broad, but we are going to continue to need to have partners that bring that complementary technology to enable us to really realise the full potential of the iBodies.
I think I just add to that. I think it's important that it is very broad when you look at it, but the actual engine of discovery in AdAlta, it's very similar, and so it doesn't mean that we're sort of focused everywhere. We know how to interrogate these libraries to try and find the best binders for a particular target, and then it's the applications of those that we can then expand, I think.
Thanks, Mick. Perhaps a question for Shaun now. So we've spoken a lot about solid tumours today, but can your CAR-T technology be adapted to tackle this as non-solid tumours, I guess, blood cancers as well?
The short answer to that is yes, as long as blood cancers have the antigens that we're targeting. We do have an in-house program that's taken a bit of a backstep at the moment because of the great results we've got with the LGR5 against colon cancer. So that's the main priority at the moment. But we do have preclinical models to look at a range of leukemias, and we always include B and T cell leukemias in our in vitro testing for our CARs. So we already have information that indicates that some of our CAR T cells at least can target human leukemias.
Thanks, Shaun. Deb, perhaps one for you. How does the decision-making process work? I'm directing this towards you because you mentioned earlier there are some specific contemplations in the agreement around this. So how does the decision-making process work? Develop further or out-license, etc., and in the event that initial assets show promise?
Well, we've taken a pretty pragmatic approach, and we formed through the actual agreement a committee that will actually take these decisions. And under different scenarios, we've already discussed how those decisions, the framework for those decisions, will be made. And so it's all pretty clear, actually, that if certain things happen, then this is the path. If other things happen, this is another path that the collaboration results will go down. And one of the things that we're looking, not everything Carina will take forward, not everything AdAlta will take forward. There's such a strong interest in licensing CAR-Ts for different types of cancers at the moment that we think that there will be opportunities to out-license rather than retain for in-house development that could be very beneficial for both sets of shareholders.
Thank you. And could you just extend on the opportunities that you see? So the size of the solid tumour market with CAR-T, as opposed to there are already 5 marketed therapies, or 6 actually on the market for blood cancers. So what does that solid tumours market look like?
Well, it's always difficult to predict market opportunity, but there are some very bullish estimates out there that have been put in place by various market research houses, and they've indicated that by 2028, the cell therapy market will exceed $20 billion US in revenues a year, and that at least 50% of that will be driven by new CAR-T therapies for solid cancers. So that gives you some idea of what the opportunity might be. But history will tell us that often these opportunities are underestimated when they're at this emerging stage, because people don't take into account the fact that they will replace some of the older therapies. And so the market opportunities as each new market entrant will grow, but also it grows because they're taking over from some of the older therapies that previously were part of the fight against cancer.
Thank you. So a question here I might just throw to Tim. How are you planning to advise the market of your CAR-T collaboration and interest the market in what you're doing?
So the first step of that is obviously the conversation we're having today. We're now at the starting line of what we think is a very exciting program. We will disclose progress as is relevant for shareholders and investors to understand the progress we're making towards the goals we've set, right? So it's a little bit hard to say exactly what will happen, when it will happen, because these are necessarily programs that develop along the way, but you can easily envisage that the early preclinical work demonstrating in vitro and in vivo proof of concept is going to be relevant and of interest to people, and that would be likely points where we'll be able to share further information. So we'll do that through our normal ASX and investor relations channels, investor conferences, and so on. We'll also continue to present our work and our company at industry trade shows and investment and partnering events, events like Bio, for example. And this is the area where we continue to engage with other companies to explore what A, their interests and needs are from a drug development perspective, and B, then how our technology can help. And we've already actually had a number of queries about whether we can put an iBody on a CAR T cell. We've necessarily to now been a little bit hesitant about that, beyond saying yes, we can do it in theory, because we wanted to make sure that we had this collaboration out there as demonstration that we could actually do it. And so we'll continue to be able to present ourselves to the growing market, and that will potentially bring new target ideas to this collaboration, or it might bring other applications of iBodies that other companies want to pursue with us. We also do know that through a previous collaboration that we can actually deliver the gene for an iBody into an animal and have the iBody expressed within the animal itself. So that's another way of actually delivering the iBodies. It's very similar to this. So that's how we're going to continue to update the market. I'll probably hand over to Deborah because Carina's still private, so they have a different process.
Thanks, Tim. And, I think it's evident through the Q&A and through the presentation how excited Carina is as well with this AdAlta collaboration. As Tim indicated, Carina is a private company. We're naturally looking to the future, and at some point we may consider becoming a public company. But for the meantime, we have our own shareholder base. We keep them regularly informed of our activities with emails and other communications that are very similar to an ASX listed entity, and we'll continue to do so.
Okay, well, with that, perhaps I might just, Tim, hand back to you for any closing comments.
Thank you, Jane, and thank you all for joining us today. This is a really exciting moment for AdAlta and for Carina, and we're really, really thrilled to be working with them. This is our second external collaboration. It's consistent with our plans to rapidly expand our iBody-enabled product pipeline. But most of all, this is about the many, many patients with solid tumours who've been looking at the success of CAR Ts in blood cancers and saying, when are they coming to us? And this is our contribution. We're very excited about the potential to really create these precision-engineered iBody-enabled CAR T therapies that are going to make a difference to the lives of the many, many patients out there with solid tumour cancers.
Thank you. And Deborah, perhaps a final word from you, and then we'll finish up.
Well, my final words is one of my beginning words in that Carina is hoping to create a future that defeats cancer with our CAR T technologies. And this collaboration is a great addition. It's a great addition to that technology platform that we can access to create the most effective CAR T therapy. And it's great to be working with Tim, Mick, and the AdAlta team on this as we move towards this objective.
Excellent. Well, thank you everyone for joining us. And with that, we might now all sign off. Have a good day.